Okay, hi everyone. I'm Angie Mikhail, and I've been leading our efforts in neurobiology and changing ecosystems at the Kavli Foundation. When I first joined the foundation almost 4 years ago, part of my role was to help identify a new scientific focus area within neuroscience that was understudied, undersupported, and kept in line with Fred Kavli's mission of advancing science for the benefit of humanity. I won't share exactly how we got to the intersection of environmental change and neurobiology, except to say that we were really inspired by the work of Eve Marder. And we were overall really intrigued by the idea that nervous systems evolved over hundreds of millions of years to help organisms sense and respond to changes in their environments. And yet, at this time, the environment is changing at an accelerated rate. To give you an example, during my lifetime, the amount of atmospheric CO2 has gone up by 20%. So overall, anthropogenic or human-generated changes to the environment are imposing stressors not experienced by nervous systems in recent evolutionary history. So when I first joined the foundation, I was able to spend a couple months exploring this topic and wrote a review paper where I outlined two broad scientific topics or entry points that were ways that the environment could be challenging nervous systems. The first of those being abiotic stressors such as temperature and pH associated with ocean acidification and transient heat waves. And I also focused on sensory neuroecology, the overall idea being that anthropogenic sensory pollution, such as noise, light, electromagnetic pollution, or others, could change the ways that organisms extract relevant information from their environments. And the overall topic was also really exciting to us because it carried with it implications that spanned outside of neuroscience as well. One example of that being ecological interactions, and also bridging even further this idea that if we had a mechanistic understanding of how nervous systems were responding to abiotic stressors or sensory pollution, we might be able to help inform predictions at other levels of biological organization and contribute to the overall environmental crisis that we're facing. And this is obviously not a very comprehensive view, but this was enough to tell us that this was a compelling and really interesting area, and that putting investments into this area could lead to really exciting discoveries that would reveal more about the natural world. So we were really compelled by what we could do, but we wanted to hear directly from the scientific community. So we convened roughly 30 interdisciplinary scientists at the end of 2022 to help inform us about what they saw as the opportunities and the needs and at what scale this work could be done. And together, the group, many of whom are in the audience today, identified two major needs or approaches that would answer this overall question of how nervous systems were responding to anthropogenic or human-generated environmental change. And the first need was to survey what is currently happening in natural populations by comparing across species, across biomes in different geographies and across multiple stressors, etc. And another complementary approach identified was to focus on mechanisms of resilience. And this could be done at sort of a smaller or intermediate scale of a single investigator or a few investigators through collaborations. And this mechanistic approach is generally the approach that the Kavli Foundation has taken. And the group also identified some ongoing needs that I won't get into detail on right now because I'm sure these will come up throughout the course of this meeting. But very briefly, there was the need to have tools to target, monitor, and manipulate neural subpopulations in non-traditional model organisms, tools for naturalistic or in-field studies, for example, portable electrophysiology devices. This one goes without saying, but also the need for interdisciplinary collaborations. And finally, some sort of standardized data infrastructure that would enable comparisons across species and different manipulations that different labs were learning about across the world. Okay, so what happened next? So after this meeting, we designed the initiative. We put our first call for proposals out and formally launched at the end of 2023. And since then, we've developed 5 active programs in addition to providing support for Gordon Research Conferences, symposia at scientific society meetings, and community events. But I'm actually not going to talk about these programs today. Instead, I'm going to take this opportunity to reflect on some of the scientific motivators that first drove our curiosity and commitment to this area. And I'll share a few examples of some of the work that we've been supporting that addresses some of the questions that we were really excited about when we first started. And the examples that I share today are by no means comprehensive, so if you are interested in learning more about work within this portfolio or other work that we fund at the Kavli Foundation, I invite you to visit our funded work page. Before I share any scientific examples, I have a public service announcement. And that is that participants of this meeting are cross-disciplinary, so it's really important to define what we mean when we use loaded terms like resilience or adaptation, and at what level of biological organization we each care about. Resilience generally is the ability to recover or the ability to maintain function despite disturbance, and it's composed of sort of 2 main parts. Resistance, which refers to the ability to withstand disturbance without significant alterations to structure or function, and also recovery, which is the speed and efficiency at which a system gets to an unperturbed state. And importantly, recovery doesn't necessarily mean that you end up with the same pre-disturbance functioning. You could end up in a different place. And if that new state of functioning somehow helps the system, whatever that system is, a neural circuit, a collection of circuits, an organism, community, etc., helps that system function better within that environmental context, that response is said to be adaptive. Okay, so one of the clear ways that climate change specifically might impact the nervous system is through abiotic factors such as temperature or pH, exerting stresses on biophysical, biochemical, or physiological processes, which function within species-specific optimal zones. And this is nicely conceptualized in Shelford's law of tolerance, which states that an organism's ecological success is dictated by its ability to maintain environmental conditions within these critical limits. And this also suggests that variability in physiological resilience across a population would be important for understanding overall species responses to environmental change caused by climate change. So some of the questions we were really excited about when we first started were, what are the neurophysiological mechanisms that confer resilience to abiotic stressors? How do these vary across time? And what are their limits? And this is especially important in the context of ectothermic or cold-blooded animals who can't regulate their body temperatures independently, but have to kind of match their internal temperatures to that of the environment. And one example of how this overall question of the limits of physiological function is approached is in the work of Dr. Florence Kerman. And in a series of experiments, her team sought to understand what happens beyond the rate of thermal tolerance. And experimentally, thermal tolerance is typically defined as the temperature at which locomotor behavior becomes disorganized, there's a loss of equilibrium, and eventually an organism will stop responding to external stimuli. So behaviorally, what you see in a little larval zebrafish is here, so loss of locomotor coordination. And if you look at the brain, what they found using in vivo calcium imaging is a brain-wide spreading depolarization, which is much like a seizure. And by manipulating the water-oxygen levels up and down, they found essentially that they could set the temperature of what this thermal tolerance was. And maybe this finding is not so surprising given the metabolic demands of brain tissue, but also what was really interesting is that all of this was able to recover within just a few minutes. So the question is, how exactly does this recovery happen? So Dr. Kerman was awarded a Kavli Exploration Award to explore specifically this recovery with a focus on astrocytes due to their known role in sensing oxygen and initiating local vasodilation to enhance oxygen delivery to brain tissue. And overall, this work will shed light on the mechanisms of physiological resilience to the abiotic stressor of temperature associated with heat waves and climate change. Okay, sensory neuroecology as it relates to changing environments is the overall concept that anthropogenic activity exerts changes to the sensory environment that organisms typically rely on to gain information about the world and guide their behavior. And you may have seen the Foundation use this image before, and I think it actually serves as a really profound example of this concept. So sea turtle hatchlings have to navigate to sea, and this has their ability to do so has a direct consequence on their fitness. And the way that they navigate and orient to sea is by using the light cue of lunar illumination and its reflection on the surface of water. And there are cases where sea turtle hatchlings actually do make it to the water, but end up turning around and going back to shore because of artificial lighting on the shore. And in this case, light pollution serves as an anthropogenic sensory trap. So light cues that the sea turtles evolved to detect and use as an information source are sort of being co-opted by artificial lighting. So the question then becomes, how do sensory systems cope with sensory pollution? And aside from thinking about sensory-guided behavioral adaptations that evolved over long timescales, like the sea turtle example, sensory cues play a major role in how organisms interact in their environments. There's many examples of this across sensory modalities. For example, noise pollution disrupting communication in songbirds, or artificial light disrupting migration in migratory birds or migratory insects like butterflies. Sensory pollution can also exert indirect effects on physiological processes. For example, artificial light at night impacting circadian rhythms and sleep-wake cycles. But it can also have direct effects like chemical pollutants modifying olfaction by altering olfactory transduction pathways. And part of the challenge of understanding the impacts of sensory pollution is actually just knowing what the sensory capabilities are of organisms that have never been studied in this context before, and then determining how that's influenced by anthropogenic sensory pollution. And that's what Kavli Grass Fellow Rosalind Putland aimed to do during her summer fellowship at the MBL. And in a series of experiments, she found that realistic levels of loud boat noise temporarily impairs hearing ability in bobtail squid. And there's some evidence in other species to suggest that temporary deafening after noise exposure is actually an active process of suppression to protect the hair cells or the auditory receptor cells from further damage. However, in the real world, this 2-hour recovery period might not be enough. There might be another boat that comes by. So now in her role, Rosalind actually uses knowledge generated from experiments like this to guide policymaking in the UK for shipping and boating routes. And this work was conducted during a summer fellowship at the MBL. And so the Kavli Foundation partnered with the Grass Foundation starting in 2022 on their flagship program, the Grass Fellowship. And since 2022, we've supported roughly 10 fellows with work focused on this topic of the intersection of neurobiology and environmental change. And several of the Kavli Grass Fellows are actually here today, and they'll be presenting throughout and chairing some of the sessions as well. Okay, so as I mentioned previously, we were initially really excited about studying neurobiology in the environmental change context because it had implications that spanned beyond neuroscience, one of those being ecological interactions. And Charles Darwin understood that interactions between species were complex or entangled, which he writes about in the last paragraph of Origin of the Species. And for the sake of time, I'm not going to read this passage, but he notes that different species and their actions within a community seem to be dependent on one another. And given that nervous systems don't exist in isolation, might that influence, if there is anthropogenic change influencing nervous systems at a large scale, might that influence the ability of a species to participate in the entangled bank of ecological interactions? And what would be the downstream effects of that? An example that approaches this concept is through understanding plant and pollinator interactions and how they're influenced by anthropogenic activity. In a collaboration between sensory neuroecologist Jeff Riffle, who's here today, and atmospheric scientist Joel Thornton. Their team found that atmospheric pollutants react directly with floral scents that are used by pollinators to locate and navigate to their flower sources. So with atmospheric pollutants, this results in a reduced ability for the pollinators to actually locate their flowers. And the team also integrated what they learned about pollinator recognition with global atmospheric models of floral scent oxidation to identify that urban areas where pollinators have a reduced ability to locate flowers. So last year, Drs. Riffle and Thornton were awarded an NSF Kavli Neurobiology and Changing Ecosystems grant to follow up on this work and to understand exactly how the olfactory system of multiple pollinator species processes olfactory information and how this processing is disrupted by pollutants to result in the overall behavioral change in a reduction of flower location or recognition. And we're really excited about this work, not only because it has implications for ecological interactions, but it also has implications for global food security, given that roughly 35% of the world's food crops rely on pollinators. And beyond ecological interactions, another really exciting implication of this body of work was that the potential to bridge what we're learning in neuroscience across scales and to inform potentially what's occurring at other scales of biological organization. In one example where I think neurobiology could really contribute to the environmental crisis is helping to better inform species extinction models, which, as I learned recently, are highly variable, predicting anywhere from 1 to 70% extinction rates in the next 50 years. So not really a helpful statistic. So if we want to bridge what we're learning in neurobiology, we obviously need interdisciplinary expertise. In support of this aspiration to bridge across scales, we partnered with who we think of as the experts in forging interdisciplinary collaborations, Research Corporation for Science Advancement. And Research Corporation is actually the oldest private foundation in the country. And they have a program called the Sciologue that brings together interdisciplinary pre-tenured faculty to annual meetings to share their work and discuss potential ways that they could collaborate. In this case, the group was comprised of neurobiologists, population ecologists, evolutionary biologists, and many others, including climate modelers. Research Corporation actually uses an algorithm to pair people together, and this algorithm has been refined over a decade. And it pairs people together that would be likely to collaborate with one another. And at the annual meeting, there's time built in for proposal development and submission, and the awarded proposals are then funded by a group of funding partners, in this case, alongside the Kavli Foundation, the Frederick Gardner Cottrell Foundation, and the Paul G. Allen Frontiers Group. So the first annual meeting was just a couple weeks ago in Tucson, and many of the fellows and the facilitators are here. And we're really excited to see what science comes out of this. My personal highlight was hearing from a climate scientist that maybe having a mechanistic understanding of neurobiology might add information about carbon cycling and actually improve climate models. So kind of beyond what I had even anticipated. Okay, so now I've shared 4 out of 5 of the active programs. I'm just really quickly going to go over the last one, which is an educational webinar series that we're putting on in partnership with the Society for Neuroscience. And the overall series is focused on this question of timescales. So each one of the webinar segments is focused on an individual timescale and overall spanning from neuromodulatory adaptation to evolutionary adaptations. The next webinar is in a couple weeks, and all our speakers and moderator are here. You'll all be hearing from them today, too. I guess I should tell you who they are. Yves Marder is a speaker, and Jan Siemens, and the session will be moderated by Laura DeVal. Importantly, SFN membership is not required. Okay, so I've now given you some examples of work that we're supporting within each of these programs. I haven't talked about how Strategically, the initiative is designed to support a diversity of funding scopes and career stages. So if anyone's interested in learning more about kind of the strategy around how this initiative is built, please do come find me during the event. Okay, so now what? Why are we all here? We recognize that the advancement of science happens through dialogue and through sharing and synthesizing research findings. So alongside our partners at the Frontiers Group, we assemble you all here today to start this dialogue. And this is the first time ever that a group of this size has come together in support of this topic. What I hope to learn from others during the course of this meeting are, what are your motivating concepts? What drives you scientifically to do this work? I also hope that we collectively challenge ourselves to consider what would be needed to advance this area of research. So what do we not know? What do we want to know? And how might we get there? For example, does the community need more collaboration with biogeochemists or marine scientists? And I say this example because I personally think that this is a huge area of opportunity that has not yet started. Okay, so what's coming in the next 2 days? After this session, where you'll hear an overview from the funding partners and also the keynote from Dr. Eve Martyr, we'll have a range of scientific topics over the next 2 days, ranging from genetics and development all the way up to ecology and evolution. Later today, there will be a group photo, also a reception and poster session. Tomorrow during lunch, there is an optional session hosted by the Allen Institute Team for Education and Engagement on engaging the public on sensitive science topics. You do have to register if you want to attend, and you can do so at the front desk when you come into the building. We also will go deeper into this topic of handling multidimensional data that's being generated from work in this space during a panel session tomorrow afternoon. Finally, we'll close out the meeting with a synthesis session on our reflections and key insights. So throughout the meeting, we encourage you to ask yourselves and each other these 2 questions that we'll use during the synthesis session at the end of the meeting. And these 2 questions are, what are you learning and what are you inspired to do next? So your answers to these questions could be about the science, they could be about the community and its needs, they could be about the growing field and where you hope that it goes in the next few years, or anything really that speaks to you about this topic or this meeting. And the emphasis here is to think about things that are actionable. For example, you could say that after this meeting, I want to learn more about some particular topic or start a collaboration, etc. The point here is that we want to be future-looking and active in the way that we're approaching this topic. Also, I didn't bring it up here, but if you noticed our napkins, cocktail napkins, if you flip them around, there's some questions on them that will help you get started. Okay, and last thing that I'll say is, in developing this initiative, I'm inspired by the early career researchers who are incredibly engaged and excited about this topic and hope to build their careers on this. So in an effort to elevate their voices, all of the session chairs will be postdocs, and they're actually going to help guide us through the synthesis session at the end of the meeting. So thank you all. Finally, I'd like to acknowledge my colleagues at the Kavli Foundation. Together we work to realize Fred Kavli's mission of advancing science for the benefit of humanity. I'd also like to acknowledge all of the support from our funders, or our funding partners, our partners, who enable us to carry out important work and help amplify the impact of the work that we do. If you're interested in learning more, you can come talk to either Amy, Stephanie, or myself, or reach out to science@kavlifoundation.org. Thank you.
Yay. Okay. Good morning, everybody, and thank you so much, Angie. Angie's really put us in a place to be successful, I think, for the next 2 days. I feel like I am ready to go, and the only thing now that stands between you and 2 days of amazing science is my opportunity to introduce you to the Frontiers Group and our mission and our footprint in this space. And I will do that, and then we will go to the science and stay there for the next 2 days. So I'm Jody Lilly, and I'm a program officer with the Paul G. Allen Frontiers Group. And the Frontiers Group mission is to encourage new ways of doing science, to nurture breakthrough discoveries, and to foster a creative community of scientists. Our team is constantly seeking novel ideas and approaches and emerging fields where an early investment could really make a difference for humanity. To carry this out, we work closely with both the Paul G. Allen Family Foundation and the Allen Institute. Our team is operationally supported by the Allen Institute, and we definitely benefit from being surrounded scientifically by all the amazing scientists and science that's here in this building. And our purpose is to advise and make good recommendations on funding for bioscience research to the Paul G. Allen Family Foundation, who ultimately make the decisions on the awards that we support and projects that we partner with. The Frontiers Group, this is our lean and mighty team. The Frontiers Group is embedded within the Office of Science and Innovation, which is led by Executive Vice President Kathy Richmond. We're 3 program officers, myself, Han Arbach, Annabelle Beichman, and our coordinator, Jessica Iverson. We very recently, well, it's not so very anymore, but we recently hired VP Julie Harris, who you met earlier today. And though Julie joined us in January, she's no stranger here to the Allen Institute. She previously worked here in brain science for nearly 10 years. And our team really takes this 3-pronged approach that consists of continuous landscaping, finding and funding bold ideas, and fostering connections to accelerate research. For landscaping, we very much rely on creative minds and generosity of people just like you that are in this room that are all over the world that share your thoughts and expertise with us. We ask a lot of questions, and we hold discussions of various formats with thought leaders across diverse fields. Once we have done the due diligence on the topic and vetted it, and it looks ripe for a funding call, we frame that open call, ask for proposals with approval from the Paul G. Allen Family Foundation. And we use 3 main funding mechanisms. The Allen Distinguished Investigator Program are 3-year awards of $1 to $1.5 million each. And each year, we typically have 2 cohorts that are organized around a theme, and we usually have about 20 active awards at any given year. The Allen Discovery Centers, which you'll hear more about throughout the science talks, are our larger awards. They're $10 million for 4 years with the possibility of renewal for a second phase of funding. We're also, as evidenced by today, we are strong believers in forming strategic partnerships. And we know that by combining forces with other funders, we can often make an even bigger impact on research in emerging fields of interest than we could do on our own. What opportunities are coming up soon? Well, we've been actively landscaping in the rapidly moving field of AI and biology. We just closed a listening campaign to solicit bold ideas from the community and to see if anything bubbled up that aligns with the Frontiers mission. And we are actively landscaping several other exciting topics, so please stay tuned. You can find information about these on our website, and so look out for announcements later this year. But first, speaking of strategic partnerships again, you heard from Angie that Kavli began convening thought leaders and in the neurobiology and changing ecosystem space. And this event that Angie referenced earlier, Kathy Richmond attended that event in 2022. And you heard about this need that came out of that meeting for large-scale coordinated efforts with the focus on comparative approaches across species, biomes, stressors, and levels of organization, and so on. And we recognized that indeed we thought this topic did align with was ripe for frontier investment, and we're really excited and thought that we could utilize our Allen Discovery Center mechanism in this space. And so our response was to open a call for the 2024 Allen Discovery Center for Neurobiology and Changing Environments, which again is that $10 million over 4 years with a possibility for renewal. We also, so we heard, I don't have to go into this because Angie did such a great job of explaining. We partnered with Kavli Foundation, Frederick Gardner Cottrell Foundation, and Research Corporation for Science Advancement on that dialogue in the Neurobiology and Changing Ecosystems. And it was such a delight to be in Tucson last month, and again, seeing so many familiar faces now from that, and very excited to see the projects that come out. So I am going to hand over to Han now to tell you a little bit more about some other initiatives.
Thank you, Jody, for the nice introduction.
I didn't
Yeah, so I am really excited to tell you about another partnering opportunity. So we've strategically partnered with NSF on a funding call, which is now closed, for microorganism-mediated organismal resilience for climate change, or MORC, if that's too much of a mouthful. And so we really wanted to take advantage of the opportunity that microorganisms have with their quick generation time, their natural abundance everywhere across the world in all kinds of ecosystems, to see maybe where there's points where resilience is conferred to microbes and their interacting partners. I'm going to go back and talk about the Allen Discovery Center now. So it's my great pleasure to introduce the Allen Discovery Center for Neurobiology and Changing Environments and its leaders, Martine Trusqueras, Amro Hammoud, and Dee Lyons, based at the Scripps Institution for Oceanography at UC San Diego. So we've heard that climate change is forcing the nervous system of many species to deal with rapid and unpredictable change in their environments, but understanding how the animals respond and adapt or not is still a mystery. And so this team proposed a really, really inspiring multidisciplinary approach to study how the nervous system of four very strategically selected marine species are impacted by current environmental changes, and what mechanisms may confer susceptibility or resilience through four interdisciplinary spheres of investigation in neural activity, neural cytogenomics, physiological and behavior neurogenomics, and environmental neurogenomics. The team has clearly articulated their dedication to open science and engagement with the scientific community. I know there's a representative from the Birch Aquarium here, for example, so you should definitely go find Megan and talk to her about that, to engage the broader scientific community and also the non-scientific community in this critical issue. So I would really like to, at this time, have you all join me in welcoming and congratulating this team on their new award, and wish them good luck over the next 4 years. And you'll be hearing many talks from the center, including from the three leaders and the other investigators. There's 13 total PIs on this award, so it's a really, really massive effort in this. And then also I encourage you to look at everyone's posters at the posters at the poster session, but there are postdocs and grad students affiliated with the Discovery Center who will have posters at the poster session this evening. So now I will hand it back off to Angie, who will introduce our keynote speaker.